Um, First, I would like to thank you all for attending the seminar, and for those that are in the virtual space doing the same thing. Before I introduce Dr. Hurley, I would like to give you some housekeeping information. So if you have a question and you're online, if you have a question, please post a question in the Q&A option, and I will read it at the end of the seminar. If you're here, please raise your hand. And at the end of the seminar, those of you that are going to have lunch with the speaker, please wait here, and I will take you to the lunch room. Okay. So today we have Dr. Jennifer Hurley as our speaker. I have known Jennifer for very many years. I've been lucky enough to follow some of her more remarkable work and meet her pretty much in every single conference that we've been together. Jennifer is a unique scientist in many ways. She's her background, and let me tell you a little bit about her background before I describe her work. She did her PhD, received her PhD from Rutgers University, and then she went for a postdoc, Dartmouth, and she worked there with Jay Dunlap in Neurospora crassa for a really long time. She did a tremendous productive postdoc. She focused mainly on the Neurospora clock, but then she moved to her independent position at the Rensselaer Polytechnic Institute, where she has moved through the ranks and now is associate professor with tenure and will be department chair very soon. I'm not kidding. She's trying to avoid that, but I'm not kidding. But what I really like about her work is that she's one of those few scientists that, despite contributing a lot to our understanding of post-translational modifications on the clock and how they work, she didn't stay in that unique niche, but she saw the opportunity of using the tools that she learned through her own career and to answer very many other problems that we have in the circadian field. She's one of the few people in the circadian field that actually is a great biochemist. Okay, you know, an area of research that is very much underrated with all the omics that we have around. But she has done tremendous contribution in our understanding of protein folding and those kind of things. She also has a lab that focuses in multiple areas, from pure biochemistry to immunometabolics. I mean, it's incredible, the spectrum of research that she's doing. Her lab has 12 graduate students, okay, at Tech. And I have only 4 graduate students, and I can't even imagine having 12, and they all have different projects. So I can imagine that her nights are much longer than my nights. But that's one thing that is amazing about her. The other thing that I want to tell you is that she's a very prolific scientist. In 6 years, she managed to publish 27 papers. And I'm not talking about 27 papers like in, those journals where whatever you publish is accepted, okay? She published 27 papers in journals that are really excellent, good quality, okay? So, so her result is, her work is not only prolific in terms of number of publications, but the quality of the publication is incredible. So as you can imagine, she won the award for the junior researcher at the Gordon Conference on Chronobiology, and an exceptional junior researcher. She has multiple awards, including an NSF Career Award. She is very well funded by NIH. Well, I mean, if you want to have 12 students, you need to be very well funded. It's kind of redundant, but I don't know. I mean, I think she's a very good role model for many of our junior faculties in the field, and I really admire your work a lot. So thanks a lot for coming and for joining us today. Okay, without any ado, I leave the audience to you.
I'll start off by thanking Carla for the perhaps overly generous introduction. To hear that she admires me is a little odd because she's definitely one of the people in the circadian field that um, I cannot say enough about how much has influenced my own career from the time that I believe she lectured me about saying no to things as an early career faculty member to also just borderlining some of the work that we do and really bringing it into the mammalian space. And so it's really an honor to be here amongst this group and to talk to you will be a bit of a high level overview of what the clock has learned about how intrinsic disorder helps to time a very highly ordered circadian system. And I know that there are some circadian people here in the audience, but for those of you who do not think about the clock at the same obsessive level that I do, just a little bit of introduction first. So if you look at a clock on the wall, you can tell what time of day it is by looking at where the hands are, right? Well, I can similarly look at your own biology and understand exactly where we are in our day-night cycle by looking at things like blood pressure and body temperature. And if you take the face off of that clock, you can see that there are a whole bunch of gears and pulleys and springs that all go into keeping that time. And we have evolved inside our own bodies a whole host of molecular gears and pulleys and springs that allow us to keep time. So why would we spend so much evolutionary energy evolving these systems? Well, because we live on a planet, whether or not you think about it every day, with a really profound night-day cycle. So we have very bright days and we have very dark nights. And along with that is changes in temperature and weather and pressure and all other things that biology has to get used to and adapt to in order to be more prepared in its own environment. And so what we have developed to deal with this is what we call circadian rhythms. So what is a circadian rhythm? Well, for those who are in the field, I'm sure you're going to tell me that there are a very strict set of rules, and I'm sure you've memorized them all and tell everybody them at your talks. But for this talk, you can forget about them. The only rule you have to know for this talk today is that a circadian rhythm is a rhythm with around a 24-hour period. Now, these rhythms are highly conserved, so we think that Earth started revolving on its axis around 4 billion years ago. So it's probably not surprising to know that we think the first circadian rhythm evolved around 3 billion years ago or so in bacteria. What's always amazing to me, though, is if you look at how these clocks work in bacteria, in flies, in fungus, in humans, they all have a lot of the same principles that allow us to keep time. And so in studying other organisms, we can understand how clocks work within our own bodies. Now, with all this regulation, you're probably not surprised when I'm going to tell you that clocks dictate an enormous amount of biology. So the very first circadian rhythm was demonstrated by Jean-Jacques de Marion in 1729, and de Marion took a plant and he put it in a box. And prior to this experiment, there had been many, many documented writings of plant movements over the day. But in putting the plant into a box, de Marion was able to show us that the plant didn't just respond to the sun, but actually anticipated where it knew the sun was going to be the next day. Now, since de Marion's initial observation, there have been a lot of things that have been shown to be truly circadian. And in my lab alone, we study fungal luminescence and metabolism and the immune response. And those are really just the snowflakes on the tip of the iceberg of what the circadian clock controls. Now, the part of the clock that always shocks me the most with all this conservation and all this regulation is that the clock is not essential. You can actually knock out the genes that run your clock, and you can even ablate the SCN of a mouse, and the organism survives. But if you ask that organism to compete in a 24-hour environment with other organisms that have circadian rhythms, that organism is going to be outcompeted every time. And my favorite example of this is the sunflower. And most people know the sunflower gets its name not because it just looks like a sunflower, but it actually follows the sun over the day. We call that heliotropism. But Stacey Harmer's group at UCSD was able to show that if you knock out the clock genes of a sunflower, that the transition from nighttime when the sun sets, when the sunflower turns back east in anticipation of sunrise, is broken. And by breaking that clock, you can reduce the amount of heat that sunflower sees every day, and reducing the heat of the sunflower reduces pollinators. And so simply by turning east to anticipate sunrise, you can increase your spread of your own DNA and give yourself a really strong competitive advantage. Now, I'm sure that you are all excellent little sunflowers. And as soon as that sun sets, that you turn off your computers and put away your cell phones and tuck yourself in bed in anticipation of where the sun is going to rise the next day, right? Wrong. We as a species are terrible little sunflowers. And as soon as that sun sets, we start treating nighttime like it's daytime. And this can have a really disruptive effect on the circadian clock. And that's one of the primary reasons that we study circadian rhythms. We ask ourselves, what happens when we mess with this process? Well, we're about to go through one of those exact experiences, and that is the shift in daylight savings time to standard time. And whenever I give my lectures, people groan because no one likes this transition. It feels miserable. Well, it's more than just feeling miserable. In just 1 hour shift once in your lifetime, we can document on a population level that there's an increased risk for heart attack, accident, and a lower cognitive output. And that's 1 hour shift once in your life. Now, if you imagine going through these kinds of shifts over your entire career, 40 years, and this is what happens for nurses and truck drivers and airline pilots, you can imagine that this miserable feeling can be amplified. And what happens in these subpopulations that regularly undergo circadian shift work? Well, in these populations, we see increased risk for cardiovascular disease, reproductive disorders, metabolic effects, mental health issues, and cancer. And in fact, the cancer prevalency in shift work is so profound that the CDC classifies shift work as a class 2A carcinogen, which is roughly the same thing as breathing in diesel fuels for your entire career. Now, if this 20% of the population was the population we were worried about, that would be a big problem. But as I just told you, we are exposing ourselves to bright light at night. And that means that we're shifting the onset of our hormone melatonin and shifting the onset of our sleep, which actually shifts the offset of our sleep and puts us out of rhythm with our regular day-night cycle. And so what we thought was an issue for 20% of the population might be an issue for a much broader percent of the people who live in the westernized world. Now, beyond what we care about when the clock breaks, we also know, as I told you, that the clock controls a lot of biology. And some of that biology are drug targets within our own systems. And so if we know what's going on and what the clock is controlling, we can identify these drug targets and maybe employ what we are referring to as chronotherapeutics to try and understand if we treat things at the right time of day, perhaps we'll have better efficacies of the drugs that we are working with. And so understanding what happens when the clock goes wrong and how we can exploit the clock when it goes right are the reasons that we really want to understand what the clock controls. So how does the clock control all of these things? Well, the clock works in a simple mechanism that's rather elegant. It is a transcription, translation, negative feedback loop. And the clock works by a two-component system where a positive arm of the clock, a heterodimeric transcription factor complex, turns on transcription and translation of the negative arm of the clock. The negative arm of the clock then interacts with that positive arm, shutting off activity of the positive arm and inhibiting the production of the negative arm. The negative arm then goes through a very specifically timed life cycle, which regulates the length of the circadian period. In doing so, the cycle can be, the negative arm is degraded and removed from the system, and the positive arm can be reactivated and the cycle can begin anew. And so it's in this way that you can have an inherent clock within a system. But this clock has to reach out to affect physiology somehow. And for a long time, it's been understood that not only does the positive arm control the negative arm, but it also controls a whole host of gene promoters that we call clock control genes, which are controlled by the clock, but do not act in the core clock mechanism. So when the positive arm is on, these genes are active. And when the positive arm is off, because the negative arm has been activated, these clock control genes are off. And so it's in this way that the oscillation of the negative arm can lead directly to the oscillation of these clock control genes. And it was actually this mechanism that was given the Nobel Prize in 2017 for understanding how the clock can control biology. However, you can see from this basic mechanism where some very common conventions can arise. And one of these big conventions is that you can tell what the clock is controlling by transcriptionally sampling all the elements over time in a cell. Now that makes one big assumption, and the assumption is that it assumes that mRNA is an accurate protein counterpoint. But we know from studies beyond the circadian field that this is in fact not true. If you look at correlations between mRNA and protein levels, you'll see that they're not always one-to-one, and in fact, they can be as low as things like you see here, which is a correlation of 0.4, which means there's some connection between RNA and protein, but it's definitely not easy to understand what RNA is doing or protein is doing by looking at the RNA levels. Now, in addition to this, in work done by someone sitting right in this room, there are a lot of examples to show that there's a whole host of things that are controlled by the circadian clock on the post-transcriptional level. So knowing this, that there's a lot of difference between RNA and protein, and that the clock is controlling a lot of post-transcriptional regulation, that's where I started my lab. And I wanted to know just how much post-transcriptional regulation was controlled by the circadian clock. So to do so, I went back to my model organism of choice, which is Neurospora crassa. And you all know Neurospora crassa, even though you've probably forgotten it, because you probably slept through the class in Biochemistry 101, where you learned that beetle and Tatum demonstrated that one gene led to one enzyme. And Neurospora is the organism that beetle and Tatum used to do that experiment. And they use Neurospora because it is an excellent genetic and biochemical organism. It also has a full knockout library, and you can make a mutation in Neurospora from start to finish in 2 weeks and have information on what you've done to this cell. And in addition, for my own work, there's a lot of beautiful genetic circadian phenotypes that we can track to understand what's going on in the clock in this organism. And we're going to talk about 2 of them today, so I'm just going to mention them now. So when we get to them, you'll understand what I'm talking about. So the 2 phenotypes that we look for in Neurospora are to see how the clock is regulating output, how it's controlling physiology, and how clock is regulating at the core level, what the core mechanism of the clock is doing. To look at output, we grow Neurospora on a long glass tube that we call a race tube. Neurospora grows on one side of the race tube to the other end, and as it grows, it forms aerial puffs of hyphae or conidia, and the formation of these puffs is directly regulated to the timing of the circadian clock. So this is the core clock regulating output. So using this race tube, I can tell you what's going on in terms of circadian regulation of output. In addition, we've also taken luciferase and fused it to the core clock genes. And then we can inoculate Neurospora under a very sensitive CCD camera, and we can track in real time the levels of core clock proteins. And so using race tubes and luciferase assays, we can tell what's going on at the core clock level as well as clock output. So knowing what we had in terms of tools in Neurospora, we decided we wanted to determine the transcriptome and proteome over circadian time to understand exactly what was being controlled by the circadian clock. So to do so, we grew Neurospora up 24 hours in the light. We then shifted it every 2 hours into the dark and age matched the samples for a 48-hour time course with a 2-hour resolution in triplicate. So this is about 75 samples worth of data. And then because we didn't want to have to pay for that experiment out of our own pockets, we reached out to the Department of Energy, which loves Neurospora because Neurospora makes biofuels. And we asked them if they would collaborate with us to sequence and use mass spec to analyze the transcriptome and proteome of Neurospora. And they surprisingly agreed. So the Joint Genome Institute at the Department of Energy took Neurospora transcripts and sequenced them over circadian time. And then the Pacific Northwest National Laboratory, which is another branch of the Department of Energy, was willing to do mass spec to understand what was controlled over circadian time. And to quantify this in a technique that I'm sure you're all familiar with, we use paired end Illumina RNA sequencing to understand what was going on in the transcriptome. And then to understand the proteome in a technique that's maybe not as familiar, we use something called tandem mass tag mass spectroscopy. And in TMTMS, what you do is you take a sample and you digest it. And then once you've digested those peptides, you fuse to every peptide in a sample a very specific mass tag. You can then mix those digested peptides that are bound to mass tags into one sample and fly them on a 2-dimensional mass spec. And using that information, you can get relatively quantitative data about what's happening to the proteome over circadian time. And so after they've done this, the DOE dumped onto our lab literally billions of data points in understanding what's oscillating in Neurospora over circadian time. And we realized we were pretty ill-equipped to deal with this problem. But fortunately, our PI has a lot of computational scientists hanging around. And what we did was to reach out to the IDEA, which is the Rensselaer Institute for Data Exploration and Applications. And they created a program for us using the harmonics oscillator equation to understand exactly what in our data set would be oscillating over circadian time. And in case anyone is interested, we actually turn this into an application that works beyond circadian oscillations. If you have cell cycle oscillations, this will find them as well. And I'm condensing about 5 years of research into one slide, of course. But using this approach, we were able to find what was oscillating in Neurospora over circadian time and assign it a p-value. And then we plotted those p-values of each RNA versus each protein. And what we found was, while there is definitely overlap, if you look down here in this corner, in the right-hand corner, there's overlap between oscillating transcripts and proteins, which we hoped we would find. We also found oscillating transcripts with no oscillating protein, but I hand waved that away with the idea that long half-lives of proteins might mask some of this oscillation. But we found that 40% of the proteins in Neurospora that oscillated with the circadian period did not oscillate at the transcript level. And so that meant that there was a lot of post-transcriptional regulation going on. But was this random or was this coordinated? Well, we went ahead and mapped all of these oscillations at the post-transcriptional level to gene ontological pathways that we have in Neurospora. And we looked at a lot of different things, but every time we looked at something, we found that these oscillations were coordinated. And my favorite example of this is here. This is the glucose 6-phosphate pathway breakdown. You can either shunt into glycolysis or into the pentose phosphate pathway. And what you're looking at here, what we've done is to map the enzymes that are post-transcriptionally regulated to these pathways. And so when you see an arrow turn orange, that's when that enzyme is peaking in its cycle. And when it's gray, that's when it's at its trough. And I hope you're noticing that the pentose phosphate pathway tends to peak antiphase to the glycolysis pathway. And in fact, all the glycolysis pathway seems to be in the same phase, while all of pentose phosphate pathway seems to be in antiphase. And so there seems to be very strong coordination at the post-transcriptional level. So this was interesting, but we wanted to see if this was in fact true across the board. So we stepped beyond Neurospora and we moved into another model system, which is here we go, mammalian macrophage. And we like mammalian macrophages for lots of reasons. Macrophages are regulators of immunity and inflammation. They're the phagocytic cells of the body, and they're very important in things like cancer and arterial plaque formation. And if this starts to sound familiar with the list I gave you of what happens when thing goes wrong with the clock, we wondered if perhaps if we could understand what a macrophage was doing, we could understand why we see disorders associated with the clock. And so that was a really nice reason to use macrophages. But in addition to all of that, we know that macrophages display robust circadian rhythms and behaviors. And we hoped that knowing that we could see oscillations, that we would see really nice oscillations in these cells. And so we did the same thing that we did in Neurospora. We asked just how much transcription can explain oscillations in our protein level. And in this case, we found that 80% of the proteins oscillated without an associated oscillating transcripts. And again, we wondered, is this just random? And we mapped gene ontological pathways. And we found, once again, that metabolism was highly post-transcriptionally regulated. And this has to be one of my favorite figures of all time, because if you look here at the TCA cycle, and I've again mapped the enzymes that are oscillating over circadian time in the macrophage. And then you look in that lower left-hand corner at the same slide of Neurospora, you can see that not only is there a lot of coordination, but many of the same exact enzymes are post-transcriptionally regulated in Neurospora as are regulated in macrophages. And this really struck us as the amount of coordination at the post-transcriptional level that would have to happen. This cannot be a random process. Now, we are not in isolation, and there are many beautiful works that have been done over the last 10 years or so to really see what we are seeing, that there's a lot of post-transcriptional regulation happening over circadian time. And so this begs to us the question, well, what is the mechanism that can impart this post-transcriptional regulation? So as I told you before, the clock is a two-component system. There's only two elements that could really put out post-transcriptional regulation. The first is the positive arm of the clock, which, as I said, is a heterodimeric protein transcription factor complex. And so it's hard to imagine besides some kind of complex networking that can be happening, that there's a lot of post-transcriptional regulation that's going to come from the positive arm. However, that second component of the system, the negative arm, we know quite a bit less about. And the one thing that we do know about is something that I discovered during my postdoc, and that is most of the negative arm proteins in the circadian clock are what we call intrinsically disordered proteins. And so let's go back to a little bit of biochemistry 101 again. For those of you who took biochemistry pre-2000, you probably learned, as I myself did, that proteins exist in a regular crystallizable conformation. And that primary amino acid sequence of a protein will dictate what that three-dimensional conformation is, and that three-dimensional conformation directly dictates its function. And that's true in some cases. But proteins, like everything else in this world, exist on a spectrum. And on the other side of that spectrum is what we call intrinsically disordered proteins, or proteins whose primary amino acid sequence does not dictate a three-dimensional conformation, but instead dictates that they exist in a heterogeneous ensemble of conformations. And what you're seeing here is what you would imagine as a well-folded, very ordered and crystallizable protein over there on your left, compared to what we envision these intrinsically disordered proteins look like. In the exact same conditions, in the exact same cell, the same amino acid sequence will dictate a lot of different conformations of these proteins. And this gives these proteins a high level of inherent flexibility. And so we wondered if perhaps the inherent flexibility was important for its function in regulating post-transcriptional output. And of course, as I said, one of the reasons that I thought this was that if you look at the predicted disorder, and here I'm showing you a plot of disordered propensity, and anything above 0.5 is predicted to be disordered within a cell, you'll see that all of these negative arm proteins in the clock are very highly disordered. And in fact, frequency, the most disordered of all of them, is predicted to be about 84% protein disorder. Now, we see disorder in the positive arm, and labs like the Parch lab and many other labs have played with disorder in the positive arm and showed that the disorder in the positive arm can change circadian timing. But there's been no evidence to show that the positive arm can regulate output when you play with this disordered regions. And one of the reasons that we linked disorder to the ability for these negative arm proteins to control biology is that in addition to being very disordered, time and time again, we see that these negative arm proteins also have lots and lots of binding partners. And in fact, if you look at a review that we published just a year ago now, you can see that if you look at the interactomes of these negative arms, these are huge complexes, up to 500 or 600 proteins interacting with these negative arm components. And so we wondered if the disorder in the negative arm could allow these proteins to be flexible to change the ensemble of interactions that they form throughout the circadian day to control output. So to answer this question, we went back to Old Faithful, and that is Neurospora crassa, to get our biochemistry work done. And in Neurospora, to give you just a little bit of terminology, the positive arm is the white collar complex, or white collar 1 and white collar 2. It turns on transcription and translation of the negative arm protein frequency, or freq. Clock biologists are very good with their naming schemes. Frequency binds to its partner, frequency interacting RNA helicase, or FRH, which helps to stabilize and shepherd frequency through its life cycle. Frequency also binds to CK1A. CK1A phosphorylates frequency until it can interact with the white collar complex. This inhibits white collar function. Frequency is then further phosphorylated as part of its life cycle until it's degraded and removed from the system, and then white collar complex reactivates and the cycle starts over again. So we hypothesized that perhaps frequency is changing conformations over the day using these phosphorylation events in order to change interactions. And we thought this could be possible because, again, as I had said before, frequency is a highly phosphorylated, intrinsically distorted protein. Studies have been done to show that frequency is phosphorylated over 100 times on a regular basis through its life cycle. And under predictions of sequence, as well as some biochemical and biophysical assays that we did while I was still a postdoc, we were able to show that frequency is highly disordered. And so again, our hypothesis is that beyond the ability of the negative arm to control the activity of the positive arm, that the negative arm changes conformation due to phosphorylation to change its interactomes, and this can eventually lead to the regulation of physiology in a cell. So this sounds like a really grand hypothesis. And then how to prove it, of course, becomes the problem. And so to understand how we're tackling this problem, you first have to understand how disordered proteins tend to interact with their binding partners. And they tend to bind through what we call short linear interacting motifs, or SLIMs. Now, SLIMs are 10 to 15 amino acid residues in length. They occur in disordered regions on proteins, and they have lots of different ways that dictate the interaction between the SLIM and the binding partner. What we are suggesting is that we are going to see allosteric changes in the ensemble of conformations of frequency, and these changes will allow SLIMs to open up and become available upon binding. But I just told you that these are disordered proteins, and it's not just any old disordered protein. Frequency is 1,000 amino acids long. So you can't NMR it, you can't cryo-EM it, and you definitely can't crystallize it. So how can you figure out whether or not you're seeing a change in the ensemble of conformations of a protein that is that big? So to do that, we got a little bit crafty. And crafty stands for Circadian Native Fast Parallel Proteolysis. And this was a project that was dreamt up by my former graduate student, Jackie Pelham. And what Jackie thought was, well, if you have a protein in a given conformation, then that protein, if you look along the length, should have regions that are protected and more available. So if you take a very promiscuous protease and you expose your protein of interest to that protease, then it should cleave at a certain rate based on its conformation because so many sequences of that cleavage site are available. Now, if you have a different conformation, then you should see a different rate of cleavage and degradation of your target protein when you expose it to the protease. And so she hypothesized that if she looked over circadian time, that she would see different degradation rates of frequency, and that theoretically would correlate to different conformations of frequency at different times of the circadian day. So Jackie did just that. She grew neurospora up over circadian time, and she exposed frequency in a native lysate to a very promiscuous protease. And she asked, do we see differences in the levels of frequency degradation over time? And if you look at the gray graph here, that is our crafty rate. Jackie showed that with regularity, we do see an oscillation in the protease digestion rate, which theoretically could be the oscillation in the heterogeneous ensemble of conformations. So step one, we do think we see oscillations in the conformation. Step two, of course, is does frequency have a diverse and changing interactome? And so to answer this question, Jackie and another graduate student in the lab, Alexander Mosier, went ahead and immunoprecipitated frequency over circadian time using some very targeted and restrictive assays. And what they found was that frequency had a very large interactome. We found about 500 or so proteins were interacting with frequency over the circadian day. We then went ahead and mapped all these interactions by time of day in some radial maps. And what we found was, of course, if you look at different times of day, you see very different interactomes forming around frequency. We also mapped the ontologies of these interactions, and we found that frequency tends to interact with proteins that are involved in metabolic processes, which hopefully you hearken back to, because I just told you that a lot of the post-transcriptional regulation that we see occurs in metabolism. Frequency? Oh, yes, we use full length frequency for this. Yep, no truncations. So when we look at the level of interactors and we coordinate it with the level of degradation rate, we can see that there's actually a coordination or a correlation between the oscillation in the ensemble of conformations and the oscillation of interactors. Jackie then reached out to a friend of ours, Joanna Chu, and what she did was to take the known interactors of frequency and actually map them based on their predicted interactors to the predicted disordered regions of frequency. And she was able to show that when we align these SLIMs, that these SLIMs occurred of our known interactors mostly in disordered regions. And then Joanna works in flies, and she gave us all the Drosophila information of PER. And we were able to see with her data that, again, excuse me, the interactions tended to occur within disordered regions. And so this gave us a really nice foundation for a hypothesis that there are allosteric changes of frequency that correlate with changes in the interactome, and that can change physiology. But as you heard me say a number of times, correlate, correlate, correlate. Correlate is great, but I want causation and I want mechanism. So how, when you've got a protein that is this size, do you drive home the idea of mechanism? So what we want to do is take one SLIM of interest, show that it's available at one time of day, and that when you change that SLIM and the availability of that SLIM, you can change the interaction and change output. Now, I'm talking about a career-long goal here. So how do we start this goal? Well, first thing we wanted to do was locate where these SLIMs interactions occurs. And you can tell that I love acronyms here. Locate stands for linear motif discovery using rational design. Our first step is really to understand where are these SLIMs on frequency so that we can tell whether or not they're more available at certain times of day. And to get after this problem, we collaborated with a friend of ours at RPI named Pankaj Karande. And Pankaj's whole goal in life is to find new ways to print peptides onto glass slides. And he cares about this because if you can do this, then you can start to figure out how antibodies interact with these types of peptides. But it struck me that he was printing peptides of about 15 amino acids in length because that was the length that we would imagine a SLIM occurring. And so we hypothesized if we could print a full map of frequency onto these glass slides, that we could flow over an interactor of interest, and then we could find where that interactor is binding to frequency. And once we know that, we can make mutations and we can zoom in on regions of interest to try and understand exactly what's important about that region of interaction, and then hopefully move that into the conformational space. Now, we had 500 interactions to choose from, but I played it safe. And instead, we decided to go for the interactor that we know always binds to, the interactor that we always know binds to frequency, and that is its partner protein, frequency interacting RNA helicase. And we call FRH the nanny protein of frequency because as soon as frequency is born, it binds to FRH. And if you break the binding between the two, you seem to increase the degradation rate of frequency and you break the clock itself. And another reason that my graduate student, Megan Jankowski, decided to use FRH is that we know a lot about the interaction between Freq and FRH already. So what Megan did was to go ahead and purify FRH from E. coli, and then she floated over a locate map of frequency. And then she first started by looking at that genetically identified region of interaction between Freq and FRH. And she asked, do we see interaction between Freq, the Freq chip and FRH itself within this particular region? And quite fortunately, we did. Everybody's great sigh of relief in the lab. The first thing we picked up was this FFD domain. However, when we zoomed out to look at the full chip of the frequency map, you can see that there's an awful lot more going on in the interaction between frequency and FRH. And Megan, being a good graduate student, didn't panic. She took a step back and said, well, what is special about these other regions? Is this just spurious binding and this isn't going to work, or do we see something special? And so she went ahead and took all these other interacting peptides and she asked us, yes. Yes.
Among these peptides, do you have overlaps on all of them?
Yeah, so they are 3-mer shift. So they're 15 amino acids long, and then we slide 3 amino acids every time when we print them. So that's why you tend to see kind of rows of them binding. Yes, good question. All right, so what did she find? Well, when she looked at the peptides that interacted between Freq and FRH, what she found about these Freq peptides is they tend to all be basic. Now, this wasn't a great surprise to us in some ways, because if you look at the FRH crystal structure I got in collaboration with Jay Dunlop and Brian Crane when I was still a postdoc, we found that the outer shell of FRH tends to be very acidic. So Megan went ahead and started mapping the interactome map to a map of the primary amino acids sequence of frequency, where we mapped the net charge per residue as you go along the sequence. And in blue, you see the positive net charge regions, and in red, you see the negative net charge regions. And Megan went ahead, and in addition to this, she mapped onto this a map of all those predicted SLIM regions that we had based on interactions. And she brought this all to me and said, you know, Jen, this seems pretty clustery, and it seems like all the interactions are occurring in these positively clustered regions. And I said, well, what do you mean by positively clustered regions? There's no meaning to me for that word. Is this significant, or is this what you see in every protein that we encounter? And she said, well, I don't know. So we reached out to a friend of ours at Washington University in St. Louis, Alex Holthaus. And Alex thinks about disordered proteins all the time. And one of the things he thinks about is charge clustering along disordered proteins. And he said, well, we can solve this problem. Let's take frequency and let's randomize it. And let's ask, when we make a random version of frequency, how many times do we see it cluster with these positive charges? And what we saw when we randomized frequency was that frequency in the native version is several standard deviations away from the average in terms of its positive clustering. Alex then reached out and pulled all kinds of frequencies out of the database. And he even went ahead and pulled all of the negative arm proteins in the circadian clock. And he found when he compared positive arm clustering to negative arm clustering, that time and again, these negative arm proteins had positive arm clusters, but no negative arm clusters. So there seems to be some evolutionary pressure to drive negative arm proteins to have these positive arm clusters. So do these positive arm clusters have any effect in terms of circadian rhythms? Well, Megan went ahead and asked that very question. And what she did was to make a series of mutations in frequency, because again, Neurospora, we can make mutants and have them ready to go in about 2 weeks. And what she showed was what you're seeing here is one triple lysine to triple alanine mutation that she made. And she asked the question, what happens to the core clock when I make this mutation, and what happens to output? And again, to get after the core clock, what she did was diffuse luciferase to frequency and ask what's happening to the rhythm. And you can see here, when we trace luciferase over time, you can see that even though the period's a little bit different, the clock is still working. There's still a running clock in the cell. However, when we grew this strain here on a race tube, you hopefully can see very easily that as compared to the wild type, which bands very nicely, which forms those hyphal kinetial formations very nicely over circadian time, that there's no oscillation whatsoever. So while the clock is running in the background, it's no longer connected to output. And this was pretty exciting for us because this is one of the first times that we've seen a functional clock without a connection to output. And it led us to think that perhaps in addition to allosteric changes that can regulate output, perhaps there's also some avidity, some electrostatically driven interactions that can help drive output regulation as well. But if you think about the biochemistry of all of this, I just told you that we made genetic mutations with just a few points in them that broke binding between freak and FRH. And if freak and FRH were simply interacting through electrostatics, then that theoretically should not be possible. So Megan went back and she looked at that region that we can break to ask, is there anything special about this particular region? And she plotted all of the peptides by their normalized binding based on their overall pKa. And what she found was that these two peptides right here, which seem to be relatively neutral, not positively charged, these are the peptides that are binding between freak and FRH in this core FFD region. She then went ahead and used the tools of locate to identify more about this particular slim region. And she showed that she could make a double arginine mutation. And in doing so, she could break completely the interaction between freak and FRH. And she took this again back into our strong biochemical organism Neurospora. And she showed that when she made these mutations, that she can actually break the binding between FRH and frequency very easily. And so this told us that not only do we see allostery and avidity in these interactions that are regulating output, but that the core clock is also regulated by slim regions that are controlled by flanking regions along this slim. So what does this give us in terms of our understanding of how the clock is controlling post-transcriptional regulation? Well, the model that we have currently is to think about frequency and FRH as a very specific type of binding partners, right? And in this binding partnership, frequency initially binds to FRH through a very specific hotspot, okay? And once that hotspot binding has occurred, frequency then can take time to recognize other regions of binding on FRH through electrostatic interactions and avidity. And so it's almost like freak and FRH come together, and then freak recognizes there's other spots it can hang on to, and it wraps around FRH in a bit of a bear hug. So how can this perhaps control output? Well, as I told you before, there's a lot of phosphorylation that occurs on frequency. And in fact, there is some beautiful work both done by the Liu lab as well as the Dunlap lab to show that there's about 100 amino acids that occur on frequency. And there's even time of day of phosphorylations of these particular amino acids. So what happens to that net charge for residue map, to those positive clustering regions, when we start to add phosphorylations to this? Well, when we look at that, you can see subtle but significant changes in these regions of net charge for residue. And you can see this a little bit easier in a map when I pick up one particular time. You can see if you look at this N-terminal end, without phosphorylation, there's a lot of positive residue charge here. When that residue is phosphorylated, or those series of residues are phosphorylated, you can see that all of this positive charge turns into negative charge. And you can, if you look at when those phosphorylations occur, they all tend to occur not at the beginning of freak's life cycle, but towards the end of frequency's life cycle. So we are imagining that what's happening in these electrostatic interaction regions is that frequency is being phosphorylated. And in being phosphorylated, you can change the interaction between freq and FRH to change frequency's conformation and make SLIMs more available for binding. Now, in doing so, this could be the underlying mechanism to our model, where frequency is not only acting in that transcription translation negative feedback loop, but changing conformation to change interactors through phosphorylation to change the output of the circadian clock. And it's important to note here that frequency, as I told you way back in the beginning, is very similar to other proteins in other organisms. And in fact, if you look at the periods in flies and in mammalian cells, you can see that, again, they have lots of phosphorylation. They have lots of positive charge complex, and we are working on right now to demonstrate that they have lots of changes in conformation. Now, if they change this conformation, what can they do in terms of regulating biology? Well, as I talked to some cancer folks yesterday, I went ahead just quickly and dug through the interactors of frequency. And in just a brief search of the interactors of frequency, we can see it interacts with FUS1, which is involved in lung cancer, dynamin, which is heavily involved in breast cancer, and 12 different kinases that directly regulate cancer phenotypes. And so what we're trying to build here is a biophysical model of exactly how changes in conformations in disordered proteins can lead directly to changes in the output of the circadian clock. There's a lot of people to thank here. We have, as Carla noted, I have a rather large lab at this point, but we have some amazing students in my lab, particularly Megan Jankowski, who you heard the most about her work today, as well as Dr. Jackie Pelham, who's a former graduate student who has moved on as a Corey Fellow at Washington University in St. Louis. We have some great collaborators in Joanna Chu, Alex Holhouse, and Pankaj Karande. And we have lots of different funding sources. I did gloss over the other side of the lab, which works on what that output does. So if you care about clocks in Alzheimer's disease, in COVID, in metabolism, the omics side of the lab cares very deeply about that. And I'm happy to talk to you about that later. And with that, I would also like to thank all of you for your attention, and I'd be happy to take any of your questions.
Okay, I never get disappointed with your talks. Okay, Daniel, go ahead.
Thank you.
So, can you tell me if there are also seen of the expressing the kinds of properties that relate to control?
So that's a really good question. So there have been some pieces of work that are being done by the Parch Lab right now to look at the interaction between regions of period and CK1A. And so they are starting to look at those as well, but the kinases you're talking about tend to have much more ordered and structural domains. So our vision is not that these ordered proteins would change conformation, but that perhaps in the change of conformation of the negative arm, like frequency, that it might bind to and make available SLIMs on that. There are a few disordered regions in the kinases, and they could be changing conformation. And the disordered field is really interested in kinases and conformational changes. Like P53 is a completely disordered protein, and it's highly regulated. So there are lots of interactions that we can envision happening, but we're taking the clock-centric view for this, for sure.
Yes, Chris.
Wonderful talk. So kind of a bigger picture question that I have is that for most biologists, when we consider circadian rhythm, the most that we think about is that there's a zeitgeber, and typically that's light. And then any animals, plants, fungus that we present, they're going to get 12 hours of light, 12 hours of dark. And that's as far as it goes. But in the real world, there's only 2 days a year when that happens, which is days we're going to peak around.
Yeah.
So every single day it's different. And so I'm wondering, you would assume that a given molecular clock, the simple version that we learn is that it's this nice precise thing that works on a 24-hour scale. But you've clearly shown that it's a lot more complex, and it may offer room for flexibility, I suppose, to respond to changes that happen. Is some of that just because of the fact that every single day the zeitgeber is changing every day, and that's how it's been for billions of years?
Right. So we think that there's there's other things that are probably going on rather than just these interactions. So for example, I was at a disordered protein conference not too long ago, and I heard a talk that talked about the disordered regions and how they can be sensors for things like calcium. And we also know that light responses can also be filtered through some of these disordered regions. So when it comes to the light response and the input of the clock in terms of light, that's probably controlled by the positive arm of the clock, which has its own set of disordered regions that could respond to light cues. But I can envision and do, because we've actually looked at the sequence and found that there are some calcium response elements in the disordered regions of these negative arm proteins. I envision that some of this flexibility would allow for sensing and conformational changes in response to sensing these. So not just electrostatic interactions between freak and FRH, but actually a region that senses calcium, it reacts to calcium's biochemistry, and that can open up a SLIM that allows the clock to modulate its behavior based on the different environmental conditions that it's seeing. So I think that disorder probably does an awful lot more than we think it does. And there's a lot more than just light that will feed into this system. Metabolism as well. There's a couple of different elements in the negative arm proteins that might respond to glucose as well. So there's a lot going on in there.
Metabolism, obviously, that's going to shift across an animal's lifespan over a year. Circadian pattern, and so that's largely, we know, dictated by life.
Yep. So there's been some really interesting studies done with a gene called CSP1 in Neurospora. And what CSP1 does is it actually regulates the promoter of the white collar complex, which is that positive arm protein. And so what it does to, it suppresses the transcriptional amount of white collar so that when high glucose levels are present and translation increases, you don't get an overwhelming amount of white collar 1. So simple changes like transcriptional rates can actually change and allow the clock to sense metabolism, but there's probably more to it than just that. There's other points of regulation that can occur, and it can occur through some of these disordered parts as well.
I have a quick question.
Yeah.
I'm very interested in mutations in part 2. So I know that the system and frequency allows you to test these mutations very easily, way much more than what we do in mammals. So do you happen to know if these SLIMs are particularly well conserved among all the pairs? And if how many of those SLIMs motifs they need to be actually mutated to dissociate a molecule? Like, for example, you mentioned FUS1, right?
Mm-hmm.
One of the molecules that is involved in cancers. So I mean, with part 2 being so unstructured and so sticky to partners, it seems to me that it has to be an awful number of mutations to truly dissociate that interaction that will end up in a phenotype that is a disease, like cancer, for example. Do you have any analysis done on mutations on your particular?
Of the SLIMs yet? So we haven't gotten started on that, but it brings up a bigger question. So the interactions between disordered regions tend to be transient and pretty weak. So we actually would predict that we're going to see not too many mutations take to break the SLIM. And the way that I think about this is somebody brought this up the other day at another talk that I was giving, and they asked the question, well, you know, you see all these minor changes. Does this actually affect biology? How can such weak interactions with such low levels of protein, because frequency and period are not high level proteins in the cell, how can you expect that to change physiology? And my answer to that question is the same thing I told you at the beginning, where the clock is not essential, right? If we break the interactions of the clock, if we break clock function, we don't break the organism. But small, regular changes over time build up to be problematic. And so what I envision this as being is a stochastic interaction engine. And that as if there's more of it around and it's more in one particular conformation, it's more than likely to bring FUS1 in contact with its kinase to affect, you know, downstream functions of FUS1. And so it's not about very strict, rigid interactions occurring at one time a day. It's about increasing the likelihood of interactions that can occur to increase the likelihood that something is going to occur at one time of day. And if you break that in one day, okay. But if you break it over a lifetime, that's going to lead to a higher risk for cancer. And so that's how I envision this model working.
Yeah, I always saw these interactions as and mutations on clock proteins, and you know, exactly like that, like they provide a fitness advantage, right?
Yeah.
That they are not critical for developing a disease,
Correct.
but they are important for the fitness and the establishment of
Yep.
the disease with the mutation. And I think that's actually what happened with cancer. And that's why I think that disrupting the clock causes a disease as long as you have the genetic background
Correct.
that favors that disease progression.
Correct.
So anyway, okay, let me check if there is anything here among our
see a hand.
online. Yeah, I saw a hand here, sorry. Allowed to talk? Okay.
I think you have to ask them to unmute.
You need to unmute. Chat.
Maybe just click the button.
Ask to unmute. Okay. Seun Imani, you have your hand, you raise your hand. If you have a question, please unmute and ask the question. I really love, yes, Silke. Oh, or you can type the question as well.
Oh, they took their hand out. Yes. So the blobs. So it has been shown that frequency at very high concentrations, not by my lab, but in unpublished data from another lab, in very high concentrations, frequency can absolutely phase separate. However, when you take most disordered proteins and you put them in high concentration, they will phase separate. So I took that to my collaborator, Alex Holhouse, and I said, well, what do you think at the actual cellular concentration levels? And he said, it's probably unlikely. Frequency is what we would call a Janus protein, where depending on the context that it is in, it will phase separate or it will not. It can do a number of different functions. And so I would predict that at the concentrations that we have, that frequency is probably not going to be what's causing phase separation. However, helicases like FRH, which also has a very disordered N-terminal region, have also been shown to force phase separation. So it could be possible that frequency is forcing phase separation in FRH. FRH is in much higher concentrations, and it would have the power to be able to do that. So whether frequency causes it itself, I think is pretty unlikely, but we actually have an experiment going right now to show if frequency can drive differences in phase separation by other proteins based on its interactions with them or based on just changing conditions in the cell itself. So yeah, we think this is part of it, but we don't think freak is the main driver of it.
Yes.
Hi, very nice talk. So there are some circadian oscillations that are clock independent, right? Clock out frequency, so transcription proteins that oscillate.
So you're talking about a clockless oscillator, a freakless oscillator, the flows, I believe those are called. And then there has been a paper published recently. It was a science paper by Reddy, suggesting that if you knock out BMAL1, that you still get freak oscillations. My expertise, I don't see strong evidence for that. And I will go after this as an omics person. So if we took our data set with our high level of analysis and triplication, and we meddle with it and make it match what some of these other data sets look like with much more low replication, we can actually insert a lot more oscillations into our data. And so there was a study done by John Hoganesh a few years ago, and what he and his team showed was that if you have low resolution and low triplication, that you can insert a bunch of oscillations and also lose many oscillations as well. So I won't say that they're not happening, because there's some nice evidence to show that the phosphorylation and metabolic oscillations that occur inside of cells are tied directly to circadian rhythms. And in John O'Neill's work, he showed that in cells that lose their nuclei, that you can see oscillations. So the core clock happens in the nucleus, obviously, because it's driven by transcription. If you lose that in red blood cells, which don't have nuclei, that you still see some metabolic oscillations. So I won't say that it's not possible. I would say that I want to see a little more data on it before I fully agree with it. But how that's all happening, I have no idea. Is there an underlying metabolic clock? You can imagine that, right? So disordered proteins are conservedly unconserved, which is a weird sentence to say, but they are proteins that arise based on the need within the system, and they tend to arise a little bit later in evolution. So you don't see disordered proteins in bacteria and archaea very often, where you see them very often in higher eukaryotes. So it's possible that the transcription, translation, and feedback loop came later and is governing some sort of underlying metabolic oscillation that if you take that TTFL away, given the right conditions, it can turn into a 24-hour oscillation. But I think more work needs to be done to prove that's in fact the case. That's a good question.
Any other questions? If there is no any other question, let's thank Jennifer for her talk.
Thank you.
Okay, all the students that are going to have lunch with her, please wait here. And I'm going to take you to the room.